Welcome to the Passionate Minds course content review. Today we'll be talking about carbohydrates and lipids and their role as important macromolecules. So the composition of carbohydrates gives it its name. So carbo refers to carbon and hydrate refers to water, so H2O. So carbohydrates, they come in chains of different lengths and they're biologically important molecules that belong to three different categories. So you have monosaccharides, disaccharides, and polysaccharides, where saccharides refers to the carbohydrate. So carbohydrates, they play a role as a source of stored energy. They transport stored energy within complex organisms. They are structural molecules that give many organisms their shape, and they play a large role in recognition or signaling molecules in biological responses. So they will provide the cell with chemical energy, and they have many storage and structural roles. Monosaccharides are the simplest carbohydrates that consist of a monomer subunit. So in this case, let's go through each of these molecules that appear here. So fructose is a 5-carbon molecule, and it is commonly found in fruit, and it's known as what you guys call fruit sugar. Glucose is what living cells use, and it's a source of energy. So for example, we see blood sugar. Galactose over here is the sugar that is commonly found in milk products. So let's look at the key difference between alpha glucose and beta glucose. So when glucose is said to be in its alpha form, the hydroxyl points downwards. So a key trick to remember here is when you do lowercase a, the tail of the A points downwards. So that's why hydroxyl's down. For the beta glucose, you see that the B over here, the tail of that B points up, so the OH group goes above, it points upwards. So that is what I use personally to recall whether alpha glucose or beta glucose has a hydroxyl group that points below or above. Finally, a key distinction between glucose and galactose is the direction of this hydroxyl group over here. So glucose and galactose are known as stereoisomers. So a stereoisomer is when you have atoms that are bonded together in the same order, but they have a different 3D organization of atoms around one of their asymmetric carbons. So you can see this in the diagram as there's a switch in the orientation of the hydroxyl that I've marked in red. So linear versus ring structure is something important to discuss when you're talking about monosaccharides, because you might see both of them in existing forms and you'll question why am I seeing these two different forms? So the reason why you see a linear form and a ring form is because a ring formation occurs when it is exposed to water. So monosaccharides, they're generally linear in a dry state, but when they're exposed to water, they form a ring structure. So the glucose monosaccharide that we see over here, it can either be in an alpha form or a beta form, and this is at random. Occurs at random because it happens whether the attack is above or below the plane of the molecule. So that's why you have something called isomerization, which refers to the ring structure formation. In terms of equilibrium, in an equilibrium concept, these forms, they exist in equilibrium with each other. So the ring and the linear structures, they exist in equilibrium with each other, but equilibrium will strongly favor the ring formation in an aqueous or water-based solution. So in solution, for example, glucose's main configuration is a 6-membered ring like we see here, and over 99% of glucose will be found in this ring formation in an aqueous solution. Now let's talk a little bit about the mechanism. It won't be relevant until you get into an advanced biochemistry class, but I'll just go through it in case you want to see how a ring formation occurs. So what happens is you're going to have the molecule that will rotate so that it's in an ideal conformation for a reaction to occur. So the oxygens over here it'll donate to the carbon in the carbonyl group. And then carbon can't have 5 bonds at once, so the double bond between the carbon and the oxygen in the carbonyl group will break, and these 2 electrons will travel to the oxygen, and then you'll form an oxygen anion. And so these electrons in the oxygen will then form a bond with the hydrogen because it is not in a preferred state. So because hydrogen can't form 2 bonds, what will happen is its existing bond with the oxygen here will break, and so you're going to be left with beta glucose. So why is this beta glucose? I discussed this in the last slide. So we see that the OH group is on the top. So what letter has a tail that points onto the top? So that is B. Therefore, it is beta glucose. There are also other types of categorizations in terms of monosaccharides. So if a sugar has an aldehyde group, which means that the carbonyl C is the last one in the chain, so like this over here, that means that it is called an aldose because the aldehyde group is in the term is present there. In this case, we see that this carbonyl group is located internally. Hence, it is a ketone group, and thus the sugar is called a ketose. So this is an aldose, and this is a ketose. Secondly, sugars can also be named according to their numbers of carbons. So in this case, we have ribose and deoxyribose, which contains 5 carbons. Those are called pentose sugars. And this molecule over here is a hexose sugar because it contains 6 carbons. Sometimes you can have 3 carbon molecules that are 3 carbon sugars that are known as trioses. And I wanted to also mention that the difference between ribose and deoxyribose is the presence of a hydroxyl group on the carbon over here. So deoxy, so without oxygen, so that's the way to remember the difference between ribose and deoxyribose. So when monosaccharides combine through a condensation reaction, they tend to form disaccharides and polysaccharides. So a disaccharide in this case is when 2 monosaccharides join together via a dehydration reaction, or also known as a condensation reaction. So in this process, what happens is the hydroxyl group from one molecule will combine with the hydrogen of another molecule, and then water will be released. And so you'll form a glycosidic bond. In this case, we see something called a 1,2 glycosidic linkage. But to generalize, you form glycosidic bonds. So we'll see common disaccharides such as lactose, maltose, and sucrose on the next slide. So maltose, also known as malt sugar, is a disaccharide that consists of 2 glucose molecules. So the linkage is referred to as an alpha 1,4 glycosidic linkage. Lactose, so this molecule over here, is a disaccharide that consists of glucose and galactose, and it is found naturally in milk. A fun fact about lactose is that many people, they can't digest lactose as adults, and that'll result in lactose intolerance. Something that you guys might be familiar about, but you can take like pills for that. But anyways, the linkage term for this is a beta 1,4 glycosidic linkage. So the reason why it's beta is because there's two beta, uh, there's a beta glucose and a beta fructose in this case. The third type of disaccharide that I'll talk about is very common. It's called sucrose, and sucrose is also known as table sugar. So sucrose consists of an alpha glucose and a beta fructose. So this time the glycosidic term linkage name will be alpha 1 beta 2 glycosidic linkage. So that refers to what bond type exists within all of these disaccharides. So what are polysaccharides? So polysaccharides are when individual monosaccharides, multiple, they come together and they form a polysaccharide. So like we said that disaccharides are two, polysaccharides are when you have more than two. So starch is one of the most popular examples of a polysaccharide. So a little bit about starch. So plants are able to synthesize glucose using light energy through photosynthesis. And then what happens is that excess glucose that gets stored in the root in the seeds is in the form of starch. So the starch in the seeds, it provides food for the embryo as the seed will germinate, and it can also serve as a food source for humans and animals who break it down using a glucose digestive enzyme like amylase. So in starch, what happens is the glucose molecules are in the alpha form. So you have alpha glucose, that is the monosaccharide that is involved here. So with the alpha form, remember that the hydroxyl group is sticking down, right? Because we saw that the tail of the A points downwards. So what happens is that these alpha 1,4 glycosidic linkages that form, they form these linear chains over here. So you have the carbon 1 and the carbon 4, thus you form an alpha 1,4 glycosidic bond. Amylose, what happens is that it consists entirely of unbranched chains of glucose monomers that are connected by these 1,4 linkages. However, we see that amylopectin is a branched polysaccharide. So you see these branches that come off over here. It's almost like you could say that without these branches, you have amylose. So you have the linear 1,4 linkages here, but you have the addition of these alpha 1,6 glycosidic bonds. So the carbon 1 and the carbon 1 here and the carbon 6, they have a linkage between them. And we also see that in this diagram over here, just you see the difference between amylose and amylopectin. You just have these branching groups here, like I've just mentioned. So now let's talk about glycogen. So glycogen is the storage form of glucose in humans and other vertebrates. So like starch, glycogen is a polymer of glucose monomers, and it is even more highly branched than amylopectin. So that is the key difference between glycogen and amylopectin. Glycogen is more branched. So whenever blood glucose levels decrease, glycogen is broken down via hydrolysis to release glucose monomers that cells can absorb and use. Additionally, starch and glycogen tend to have helical structures, causing it to be insoluble in water. So let's talk about cellulose. Cellulose is a major component of plant cell walls, which are rigid structures that enclose the cells. So that's what makes your lettuce or other veggies crunchy. Wood and paper are common examples of items made of cellulose. And cellulose itself is made up of unbranched chains of glucose monomers linked by beta 1,4 glycosidic linkages. So like I just said, Cellulose is made up of glucose molecules, monomers in their beta form, and hence it'll give it a different property. So in this figure that we see over here, uh, every glucose monomer in the chain is flipped over in relation to its neighbor, and this will result in a long and non-helical chain of cellulose. So these chains will then cluster together to form these little parallel bundles that are held together by hydrogen bonds, and that's what will give cellulose its rigidity and high tensile strength, something that is important to have a durable and strong plant cell. So in terms of humans, the beta-glycosidic linkages in cellulose cannot be broken by human digestive enzymes, so humans are not able to digest cellulose. However, cellulose is an important component of our diets because it passes through us as undigested and insoluble fiber. It acts as roughage. But for some herbivores, like cows and buffaloes, they have microbes that help them process cellulose. So these microbes live in the digestive tract and will help break cellulose down into glucose monomers, which can be used by the animal. We also see examples of wood-chewing termites that also break down cellulose with the help of microorganisms that live in their guts. So cellulose is specific to plants, but polysaccharides also play an important structural roles in non-plant species, and this is through chitin. So arthropods, such as insects and crustaceans, they have a hard external skeleton called the exoskeleton, which protects their softer internal body parts. So this exoskeleton is made up of the macromolecule chitin, which resembles cellulose, but it is made out of modified glucose units that contains a nitrogen functional group. So chitin is a major component of the cell walls of fungi, which are neither animals nor plants, but form a kingdom of their own. So lipids are another macromolecule that is important to discuss. So let's talk about general lipid structure. So I'll just draw a triglyceride as an example. So you have a little coat hanger-like structure here called a glycerol, and then we have 3 fatty acids that branch off. So there's 3 main categories of lipids, and we'll go through each one of them in depth in the next slides, but you have triglycerides, you have phospholipids, and you have sterols. So there's a lot of roles of lipids just because of the fact that it contains so much more energy than a polysaccharide. So here's a stat for you guys to know. So a gram of fat stores more than twice as much energy as a gram of polysaccharide. So that means that a major function of fats is for energy storage in adipose cells. So it acts as a source of stored chemical energy because those carbon to carbon and carbon to hydrogen bonds has a lot of potential energy. It helps to transport stored energy within complex organisms. It acts as structural molecules that gives the organisms their shape. It even acts in recognition or signaling molecules in biological responses. And finally, in adipose tissue, what you call fat. So excess carbohydrates, they're converted into lipids and stored as fat droplets in the adipose tissue. Fat molecules are also called triacylglycerides or triglycerides, and they're primarily stored in specialized fat cells called adipocytes, which make up a tissue known as adipose tissue, which I discussed in the previous slide. So like I said before, a glycerol is a small organic molecule with 3 hydroxyl OH groups, while a fatty acid consists of a long hydrocarbon chain attached to a carboxyl group. So we see the OH group over here, and then we see the carboxylic acid over here for the fatty acid tails. So to make a fat molecule, to make a triglyceride, the hydroxyl groups on the glycerol backbone will react with the carboxyl groups of fatty acids in a dehydration synthesis or condensation reaction, And this will yield a fat molecule with 3 fatty acid tails that are bound to the glycerol backbone via something called ester linkages, like I wrote over here. So these are linkages that contain an oxygen atom next to a carbonyl group, or a C double bond O that we saw over here. So this over here, this bond, these bonds here, these are ester linkages, or ester bonds. Triglycerides may contain 3 identical fatty acid tails or 3 different fatty acid tails with different lengths or patterns of double bonds. So that means that they can different length or their degree of unsaturation. So let's say we have only single bonds present within the fatty acid tail. So that is what you call a saturated fatty acid, meaning that hydrogen atoms are attached to the maximum spots. So there's the maximum amount of hydrogen atoms present. Now, when the hydrocarbon chain has a double bond, that means that the fatty acid is said to be unsaturated, so it has fewer hydrogens. So if there's only one double bond in a fatty acid, that means it's called monounsaturated. But if there are multiple double bonds, that means it is polyunsaturated. Saturated fatty acids, tails, they tend to be straight. So this means that because they're more straighter, they can pack more tightly with one another. So this tight packing is the reason why fats tend to be solid at a room temperature, and they'll have a higher melting point. So this is why butter tends to be solid, but it's because that butter has more saturated fat. But in contrast, you have cis unsaturated fatty acid tails. They are bent because of the cis double bond, so it makes it harder for fat molecules with one or more cis unsaturated fatty acid tails to pack tightly. So fats with unsaturated tails tend to be liquid at room temperature, meaning that they have a lower melting point, and this is what we refer to as oils. So olive oil is made up mostly of unsaturated fats. Trans fats, however, are rare in nature, but are readily produced in an industrial process called partial hydrogenation. So in this process, what happens is hydrogen gases pass through the oil. So this oil is made mostly of cis unsaturated fats. And then what will happen is that the presence of hydrogen will react with these unsaturated fats, and it'll convert some, but not all of these double bonds to single bonds. But the issue with this is that trans fats have negative effects on human health, because there's a link between trans fat and coronary heart disease. Phospholipids are major components of cell membranes. They have 2 fatty acids that are attached to glycerol. So we see the 2 fatty acids over here, and then we see a glycerol over here. Generally, in the 3rd position, you see a phosphate that bears a negative charge, and that is attached to the 3rd position of the glycerol molecule. And then attached to the phosphate is a different modifier. You could have either a choline modifier, or you could have a serine modifier. So this will give the phospholipid 2 different names. You could have a phosphatidylcholine or a phosphatidylserine. So different modifiers that are attached to the phosphate group will give the phospholipid different properties and roles in a cell. Now, what happens when you interact, when the multiple phospholipids interact with water? So it's a really complex interaction. So first, let's talk about hydrophobic and hydrophilic. So you have the heads of the phospholipids are hydrophilic. So the reason why it's hydrophilic is because of the phosphate containing group bears a negative charge, and therefore it will interact readily with water. So it's polar at that part. You have a hydrophobic fatty acid tails, which come together because it's hydrophobic because it's very nonpolar. So the reason why it's nonpolar is because it contains mostly carbon to carbon and carbon to hydrogen bonds. Now, because there's a difference in polarity within this molecule, you call this amphipathic. So now let's talk about the interaction more in depth. So the hydrophobic tails will aggregate to the center and it will expose only the hydrophilic heads to create the lipid bilayer. So this is what creates that spherical structure of the cell. So when they're added to water, what will happen is they'll aggregate with the hydrophobic tails pointing towards the center and the hydrophilic heads on the outside. So this general structure is known as a micelle. And this is a major component of cell membrane. So that's why you have let's just say this is the extracellular environment. This is the intracellular. So it will help to differentiate. It'll form a barrier between the cell and the external environment. Waxes are another biologically important category of lipids. It covers the feathers of some aquatic birds and the leaf surfaces of some plants because of its hydrophobic properties, so it's water repellent. So this prevents water from sticking to or soaking into the surface. So this is why we see that water is beating up on the leaves of many plants and why birds don't get soaked when it rains. So you see how water is beating up on the feathers of this bird. So steroids are lipids with a carbon skeleton consisting of 4 fused carbon rings. So we see steroid skeleton over here. There's 4 carbon rings that are present. So different steroids can be created by varying the functional groups attached to the rings. So there's various functional groups which differentiates cholesterol and cortisol. So although they do not resemble the other lipids structurally, steroids are included in the lipid category because they're hydrophobic and insoluble in water. All steroids have 4 linked carbon rings and several of them, like cholesterol, also have a short tail on carbon 17 in cholesterol. Many steroids also have an OH functional group, which gives the classification as an alcohol. Thus, we call these steroids sterols. So now let's talk about cholesterol. So cholesterol is an important example. It's a precursor from which all other steroids are synthesized. So many of these other steroids are hormones, including the vertebrate sex hormones like testosterone. While cholesterol is clearly an essential molecule, though, high levels of cholesterol in the blood can contribute to cardiovascular disease. It is also important as a structural component in the animal cell membrane.